Another process which you routinely use for DNA analysis is electrophoresis, okay? And can we do that process on a microfluidic chip? Because if we can't separate out our reactants, we won't be able to realize, the so-called lab on a chip device. So the way to do that is that you use electrokinetic transport. So let's see what is electrokinetic transport, and then you would be able to understand how we can do DNA analysis or let's say protein separation on a chip. So consider this, in this schematic I am showing a microfluidic channel which is typically 10 microns or 10 to 100 microns thick. So every surface actually has some charge on it, okay? So in macro scale we don't feel that charge because the body forces are more important than surface forces. So when you reduce the size of your channel, the surface forces become more prominent, okay? So that is another advantage of microfluidics, or another way in which by miniaturization the physics changes. So if you take this micro channel, it has some negative charge. For example, in glass, if you put glass and take a glass capillary and fill it with water, the silanol groups will donate a proton and acquire a negative charge on the surface. So in fact, glass, you would be surprised, is actually a strong acid. It has a pKa of 5 or even less. Okay, so glass is negatively charged when in contact with water. Now let's say you have this capillary, glass capillary with a negative charge, and you fill let's say sodium chloride solution in this capillary. So you have this negative charge, it's going to attract some positive ions from the solution. So you get a zone, a very narrow zone near the surface where you have more number of positive charges than the negative charges. This is called the double layer, and the double layer is actually really small compared to the channel dimension. So it's about 10 nanometers for all, 10 nanometers and even lower for all practical purposes. Okay, so this is what happens if you fill a capillary with an electrolyte solution. Okay, so there's no additional actuation that you are doing at this point. Now let's apply an electric field. So you dip these two ends of capillary in, let's say, some beakers and apply electric field by dipping in electrodes. So what will happen is that as you would intuitively be able to justify that these positive ions are going to move due to the electric field. Okay, so when you apply electric field, these positive ions are going to move, and as they move, they'll drag the fluid along with them. So what you'll end up is getting fluid flow without having any external pumps. You just apply field, there's no moving part, and you will have fluid flow. So this is called electroosmotic flow. The flow velocity is proportional to the electric field. Okay, and electroosmotic flow has certain advantages over pressure driven flow by, let's say, apply by using external pumps. Okay, so this is what happens when you apply electric field in microchannels filled with electrolyte solution. And in addition, you would know that positive and negative ions would also migrate under the effect of electric field, and this is called electro migration. So electro migration is that if you apply electric field, ions with different size and charge are going to move at different speeds. Okay, so based on this principle is how we can do DNA separations. So using a combination of electroosmotic flow and electro migration, we can do DNA analysis, for example, on a microfluidic chip. And the advantage of doing this on a microfluidic chip is that the dimensions are really small. So you can do capillary electrophoresis in long capillaries, and that is how it was done traditionally. But let's say you have a meter long capillary, then you would apply about 10 kilovolts of voltage, okay, to get an electric field of 10 kilovolts per meter. But if you're doing it over, let's say, a small dimension, let's say 10 centimeters, you would need 10-fold less electric field. Or in particular, if you apply same electric field over shorter length, you are going to get very faster analysis, and that would reduce the analysis time as well as the precision by which you can do this analysis. So with this, I would like to show you how electroosmotic flow differs from a pressure driven flow. Okay, so anyone trained in basic fluid mechanics would know that if you apply a pressure, you will get a parabolic profile, because the walls of the channel, okay, at the walls of the channel the fluid is not going to slip. Okay, so you get a parabolic profile. Now if you have a sample analyte which you are trying to transport from one point to the other point in a microfluidic channel, and you use a pressure driven flow, you will see that the solute which is closer to the center of the channel is going to move faster than the solute which is closer to the wall. So you get so-called dispersion, and you don't want these, wide bands in your analysis. So with electroosmotic flow, you get a plug shape profile, because it's just like two walls which are moving and dragging the fluid along, and you get a very uniform profile. And this is really important in capillary electrophoresis on microfluidic chips. So with that, let's look at what is how you can, let's say, separate a sample mixture into its constituent species. Okay, so those who are not aware of electrophoresis, so it's like let's say you have a water contaminated with some pollutants, and you want to quantify, which pollutant is present in how much quantity. Okay, and you have a mixture of, let's say, pollutants along with other ions. Okay, so the first step is that let's separate out this pollutant from the other constituent ions, and then see how much it is, because when you have this mixed sample, it's hard to detect. Okay, so this process you can achieve by doing electrophoresis, which again is based on the differential electro migration speeds of ions, okay, when they move under the effect of electric field. So every ion has a different size, okay, and it's different charge. So when you apply electric field, they move at different speeds, and you can use this property to separate out these sample ions. So the way we do is that, and this is the convention, and in conventional electrophoresis also the process remains the same. So you have a background electrolyte which you maintain at a particular pH depending upon the application. Okay, so it's shown in this diagram, concentration versus the axial direction, the axial coordinate. So you have a large amount of background electrolyte, which for example can be sodium acetate, okay, if you want to maintain a particular pH, and then you have these sample ions. In this case I am showing S1 and S2, which are present at very low quantities. Okay, so if you apply electric field, okay, you know that if the sample ions are present in small quantities, okay, so they are not going to affect the resistance of this channel. So most of the current is carried by the background electrolyte. So when you apply a field, these sample ions are going to move at speeds which are proportional to their electrophoretic mobility, okay, and they separate out, and in process they also diffuse, okay, because you can't prevent them from diffusing. So eventually you get two peaks in this case, and you can measure, let's say if you are using some fluorescence based detector or UV absorbance based detector, you can measure the area under the peak and quantify the concentration of these species. Okay, but to quantify using, let's say, UV absorbance, separation is the first process that you need. Okay, now how can we do this on a microfluidic chip? Okay, so the way to do is that we use both electro migration and electroosmotic flow. So we take a chip which has a cross geometry, something like shown in this slide. So we fill this microfluidic chip with a background electrolyte, which is shown in the gray color. Okay, and in the top reservoir we'll replace the electrolyte and fill it with the sample which we are interested in analyzing, or you can mix that sample with the electrolyte itself. Okay, and in this device we have made such that the voltages at ends of this chip, or the ends of the channels, can be controlled individually. We have this chip in which we can control the voltages, and at the end ends of this channel. So the first step is to inject the sample, okay, so that we have conditions where you have a narrow band of samples which would we would like to separate it out. Okay. So we apply these voltages such that the electric fields are in this direction. So the electric field points from top to bottom and from the sides. Okay. So due to electroosmotic flow, you will have a flow which comes from this side, okay, and flow which brings in the sample from the top well down to the bottom well. Okay. So in this horizontal channel, we have a very narrow band of samples now. In the next step, let's switch the voltage such that voltages at the ends of the channel such that the electric field now points in the horizontal direction in the or in the points towards the right hand side in the horizontal channel, and then you would separate out these sample ions. So the first step is to create those conditions where you have a narrow band of sample, okay, because if you don't have a narrow band of samples and you have a broad band of samples, then you would have to travel a longer distance so that they can separate out, the sample species can separate out. So you can do it on a microfluidic chip. Here is a video showing that. So in this case, the detection is by fluorescence. So you can see these are two fluorescent species. First you inject these species and then you switch the field in a way that separation takes place. Okay. So typical time that you take for separation is about 3 minutes. So if you want to do the same experiment in traditional devices, it would take half an hour. Okay. So this is how you can do electrophoresis on a microchip, and people have done DNA sequencing on a microfluidic chip, and those who are familiar with this lab device called bioanalyzer. So this is the process that Agilent Technologies bioanalyzer actually employs. So it does microchip electrophoresis. Okay. So you've seen a way in which we can use electric field to separate out different ionic species. So for DNA analysis, actually you put a sieving matrix, okay, it's a polymer which entangles with the DNA fragments and reduces the speed of those DNA fragments based on the size. Okay. So you can do DNA analysis on these chips. But the disadvantage of this technique is that there's no way you can control or you can prevent these species or the bands from diffusing. Okay. So if you want to, let's say, analyze really low amounts of species and they are going to diffuse further, your sensitivity is going to go down. So one way you would think is that, you know, let me concentrate this sample and then let's do electrophoresis, and of course you will achieve that. And I'll show you a way by again by electrokinetic transport, you can actually achieve this so-called pre-concentration and separation all on a single device. So we use a technique which is again an electrophoresis technique called Isotachophoresis, I'll call ITP. So it's a nonlinear electrophoresis technique. In this case, we don't use a single electrolyte, we use so-called discontinuous electrolytes. Okay. So we have an electrolyte which is filled in the channel, which we'll call leading electrolyte. We then put the sample ions, and behind those sample mixture we'll put a trailing electrolyte. And these electrolytes are chosen such that the co-ions, that means the ions which have same charge as the sample ions, have mobilities such that the sample mobility of sample ions is sandwiched between the mobilities of the leading ion and the trailing ion. So what happens is that now if you apply electric field, the leading ions move very fast, okay, but it cannot you can't have you cannot break the continuity of current. So it can't happen that the leading ions have moved further and sample ions haven't moved further. So in this process, the concentration of the sample ion changes in a way that the speed of both all ions are same, okay, even though their mobility is different. And to do that, that means you need to change the conductivity. So the whole process works in a way that the conductivity or the sample concentrations gets adapted in a way that the speed of ions is same. So if you start with a very high concentration of the leading electrolyte, starting with a very low concentration of sample ions, you will increase their concentration. So the mass remains conserved, but you actually compress those samples in a very narrow zone. Okay? So you can concentrate, and starting with very low amounts of ions, if you can concentrate them in very narrow regions, you can concentrate them. Okay? And those who are interested in more physics of this process, actually the zone interfaces, the interfaces separating these two zones are actually shock waves. Okay? So shock waves are those waves which self-sharpen over time. So this is the way you can control these species from diffusing into individual zones. Okay, so this technique you can use for pre-concentration, and recently there is a startup in Silicon Valley called Purigen Biosystems who are using it for sample preparation, and I'll explain you how. So before that, let's look at this figure over here. In this case, initially low concentration of dye, which you can see as faint fluorescence in the TE zone, is being concentrated in a very narrow region, and you get very high signal. Okay? So you can get up to million-fold increase in concentration by doing this process. Again, everything happens in few minutes. Okay? So not only you can concentrate these species, you can also selectively concentrate. So that means let's say you have some DNA fragments with a particular mobility, and you choose your Le and Te ions, the leading and the trailing ions, in a way that they would only enable focusing of those DNA fragments. So if this inequality for sample other ions is not obeyed, then they are not going to get concentrated. So that means you can preferentially concentrate certain ions. So that is exactly what this chip of Purigen is doing. So they are extracting DNA from biological samples based on this process. So for sample purification, once you have purified the sample, you can use it for some downstream process. So this technique of ITP can enable, as I told you, million-fold increase in concentration of ionic species, but it doesn't do well in actually separating them out. You can separate them out, but the resolution is really low. So what we can do is we can couple this process ITP and capillary electrophoresis. So that means you pre-concentrate your sample and you can then do a separation. So ITP will give you high sensitivity, and zone electrophoresis or capillary zone electrophoresis is going to give you high resolution, in the sense that you will be able to differentiate these zones after they have separated. So in this experiment which we did, we had about 1 picogram of nucleic acid over here, and to give you an idea, that's about 1/3 of the nucleic acid content of a single cell. So in this experiment we have a 10 kilobase pair DNA ladder, which we are visualizing by an intercalating fluorescent dye. So initially we focus all those DNA fragments in a very tiny region over here. So if you concentrate them, you'll get a better fluorescence signal, and then we can initiate a separation and you can detect them. So here's a video of that. So initially this focus sample is moving, and then further downstream you can see that the separation process has initiated, and towards the end you can detect all the fragments of this DNA ladder. And again, I would like to emphasize that the initial amount of sample was only 1 picogram. So this low amounts of sample and doing electrophoresis is unheard of if you are doing gel electrophoresis. So you can imagine that the cost reduction in doing this kind of analysis on a microfluidic chip, because we are using reduced volumes. Okay. So lastly, I'll come at another talk about another application of this field. Okay. So those in biology or biotechnology would know of this probe called a molecular beacon. Okay. So let's say if you have a single standard DNA target and you want to see that if a particular sequence is present in that target or not, and you know the sequence which you are actually looking for, you will make a probe, a DNA probe, such that it has a sequence in a part of this probe. You will have a sequence which is complementary to the sequence that you are targeting, and there are some additional zones in this probe such that at the ends of these fragments you have a fluorophore and a quencher. That means one molecule gives fluorescence, okay, when it absorbs light, and the other one actually absorbs its fluorescence. So in the unbound state, this molecular beacon doesn't give fluorescence, and when it binds with a target having that particular sequence, it will give fluorescence. Okay, so it's kind of a reaction which will emit fluorescence. You can think of that way. Now, if you have a very small concentration or low concentration of DNA target, okay, and you know that if this reaction obeys second order kinetics, it's going to take very long time. So about for these reactions, let's say if you have a picomolar amount of DNA to get a particular value of fluorescence signal or reaction, or to complete the reaction, you can take about 5 days to complete this reaction. But what we do is that we can focus the molecular probe, the DNA probe, and the DNA target using ITP in a very small zone. Now, you know that if you increase the concentration of both target or both reactants, in this case the target DNA and the DNA probe, the kinetics are going to accelerate. So, imagine if you increase the concentration 10,000 fold, you will accelerate the kinetics significantly. So, once they are in ITP zone, you can have this extremely fast reaction, and you can then initiate a separation to separate out bands for the target DNA bound to the DNA probe, which is fluorescent, and the unused DNA probe. So, you need a separation eventually to see, you know, what is the concentration of the target that is bound with the probe, and which how much amount of probe is actually unbound. So, in this experiment, you can see that on the x-axis we have the axial coordinate, on the y-axis, the vertical axis, we have time coordinate. So, over time you can see that the two zones emerge. One is for the hybrid target and probe, and other is the unreacted probe. So, what would take you 5 days to complete, you can do it in 3 minutes using this technique, just because you are increasing the concentration of both reactants significantly. So, this brings me to the end of this lecture. So, I hope I have been able to communicate the advantages of using small volumes at small scales. So, there is an interesting physics that goes on at small scales, which you can use for doing your routine biotechnology experiments. And the advantage, as you would have figured out by now, is that we are using nanoliters of volume. So, you can imagine the cost reduction and the reduction in time, because the length scales are small over which we are doing these processes. And in addition, we have this opportunity of integrating large number of processes on a single chip. And I have showed you certain commercialized products based on these advantages of microfluidics. With this, I would like to thank you for your kind attention.